Welcome to the 10th anniversary of the McGovern Institute. So, the McGovern Institute. It all got started with the vision of Pat and Lori McGovern to create the McGovern Institute for Brain Research at MIT. That led to the construction of this wonderful building complex, and which opened 5 years ago. In fact, we had our opening celebration almost 5 years today, and I recognize many people here today were at the opening celebration 5 years ago. Since then, the McGovern Institute has been in operation a total of 10 years. So this would be a good time for us to reflect on why are we here? What are we trying to accomplish? And what sort of progress are we making? That brings us to the mission of the McGovern Institute, which is a twofold mission. The first is to understand the mind and brain, and the second is to apply that knowledge to understand what goes wrong in a variety of serious brain disorders. This is truly an astronomical problem. There are as many neurons in our brain as there are stars in the Milky Way. And it's the interaction of these billions and billions of neurons that lead to who we are. It leads to the human mind. It leads to our shared thoughts and dreams and aspirations. It leads to the ability to learn and remember. It leads to our innovation and creativity. Unfortunately, it also leads to conflict and sometimes leads to reconciliation. And when it goes wrong, when the processing goes wrong, it can result in a serious brain disorder. And statistically, 1 in 4 families in this room will have a brain disorder, will be affected by a family member with a brain disorder of some sort. This could be a psychiatric disorder, a disorder of childhood such as autism, a disorder of the elderly, Alzheimer's, dementia. And I don't need to tell this group that the treatments we have for these disorders are woefully inadequate. But it's not my goal here today to depress you, but rather to tell you why we have so much hope about progress over the next decade. And our hope springs from what I would say are 3 revolutions that have taken place in science in recent years. And those revolutions have been in genomics, in systems neuroscience, and in human brain imaging. And they have depended in part on very significant advances in technology. So, how are we making use of these new opportunities to drive progress here at the McGovern Institute? Well, first, as I told you, we're pursuing a dual-fold mission, which is to support basic research as well as research that we call translational, or with an eye towards explaining or helping in a serious mental disorder. And we put this all together in the same place. Secondly, we encourage what we call a culture of entrepreneurial research, so that using support from the McGoverns and from our donors, we can fund seed projects of faculty members and students who have novel new ideas, need something to get it off the ground, but are too risky for traditional funding sources. And we encourage a culture of collaboration, both within the McGovern Institute and across this wonderful building with our friends and colleagues in the Pickhour Institute and the Department of Brain and Cognitive Science, there's no more collaborative center in the world. The collaborations also extend throughout the Kendall Square area to our colleagues across at the Whitehead Institute, the Broad Institute, throughout the Boston area with the Boston hospitals, the universities throughout Boston, and even throughout the world. We have colleagues on the other side of the planet. Of course, none of this would be possible without our faculty members. Phil Sharp, who was the founding director of the McGovern Institute, assembled a group of MIT faculty members who were absolutely stellar. These were the world leaders in their field. And over these years, they have had significant accomplishments and recognition, including, of course, the winning of the Nobel Prize by Bob Horvitz 2 years after the founding of the Institute, the winning of the National Medal of Science by Anne Graybiel, and many others. And at our opening celebration, I was able to tell you about some of the accomplishments of our founding faculty members. This founding faculty has overseen the recruitment of a large number of new faculty into the McGovern Institute. And it's the work of the new faculty members that I'm going to talk a bit about today. These are faculty members, many of whom would not be here if it weren't for the McGovern Institute. Now, there's too much work to tell you about everything they're doing, so I'm just going to pick a few stories, and you'll see that a common theme of the work that I'll be presenting is a need and desire to understand what we call a neural circuit. I will start with my close collaborator, Ed Boyden. Ed, when he was at Stanford University, along with a few others, developed a revolutionary technology allowing us to control brain activity with light. And it has continued with these discoveries here in his work at MIT. This was a technology that I am honest with you, we did not dream about 5 or 6 years ago. It wasn't even on the horizon as a possibility, and now is sweeping through neuroscience labs across the world, changing how we do our research. The basic idea is that you take a light-sensitive molecule from an organism like an algae, and you use gene transfer technology to get that into neurons, so now neurons become sensitive to light, and using light, you can turn them on and off. In the living brain, you can target individual neurons, individual types of neurons. You can use different wavelengths of light to target different types of cells, turning them on and off. And ultimately, we hope to be able to control the activity in neural circuits the way that a conductor would control the musicians in an orchestra. Now, of course, this requires a neural conductor of sorts, and Ed has been collaborating with engineers and others to develop very sophisticated fiber optic arrays for controlling activity, and this has been applied in animal studies so far. But there are also some immediate therapeutic applications of this technology. Ed and his collaborators have addressed the problem of blindness caused by degeneration of the photoreceptors, which, as we all know, can occur after macular degeneration, retinitis pigmentosa, and so on. And what they recognized was that if the photoreceptors have been lost due to disease, that there's all these other neural elements in the retina that communicate the information from the photoreceptors back into the rest of the brain, and that perhaps if they could put these light-sensitive molecules into those neurons, then they could become sensitive to light, bypassing the photoreceptors. And indeed, in work that has now taken place in mice, it's been very encouraging. If you, let's see if I can get this to work, yes. If you put a normal mouse, sighted mouse, into a maze with the light, they will immediately swim towards the lighted arm. But if you take a mouse that's had its photoreceptors damaged, it will not, as you see here, it doesn't go towards the light. But if you give the same mouse essentially gene therapy, to get this material into its retina, that's shown here, you can see that the mouse immediately swims to the lighted arm. So these are early days, but they're very encouraging, and work is progressing towards eventually testing this kind of technology in people. Now, the retina is, by comparison with the rest of the brain, is a relatively simple, straightforward circuit. The kinds of circuits that would be, say, involved in a complex disorder like schizophrenia, mania, Parkinson's disease, these are very complex circuits. And without an understanding of those circuits, then we don't have too much hope of being able to control them through light or any other technology, including drugs. We need a basic understanding of how those circuits work. And that's why the neural circuit has become a focus of a lot of our work. Kai Goossens, for example, is a faculty member studying the neural circuitry underlying fear, anxiety, and depression. Jim DiCarlo is studying the neural circuitry underlying the recognition of objects by sight. Michael Fee is studying the neural circuitry that allows birds to learn and produce song. And Michael's story, I think, is a good example, and I'm going to spend just a few minutes describing how he's gone about to understand these circuits. The bird is a wonderful model for studying the neural basis for the production of complex motor sequences, in part because the bird's song is highly complex, as you can guess from the sonogram that you see here, but is highly reproducible. Let's see if I can, two examples. Those are all the syllables of the bird songs, which always come out in a very, very regular way. Michael and the people in his field have mapped the circuitry in the bird that's important for the production, learning of these kinds of bird songs. And Michael's work recently has identified the structure up here known as HVC as a regulator of the pace of complex motor productions in speech. And the reason he knows that it's important in regulating the pace of the output is because he can speed up or slow down the neural activity in HVC using temperature, and speed up or slow down the production of song. So when the HVC is warm and activity is fast, the song comes out quicker. And when the HVC has been cooled slightly, the song comes out slower, not unlike how the organ grinder regulates the production of the speed of music coming out of the organ by turning the crank. Now, it turns out it's not just the speed of sound that's regulated by this structure, it's the actual sequence of syllables that's being recognized, going from one to the next. And it turns out that this occurs in a remarkably straightforward way, in that this chain of syllables that are produced by a chain of neural activity going from one neuron to the next across the structure. And as you go through this chain, then the string of syllables comes out in the correct sequence because of the connectivity of these neurons. Not unlike the way it would be that if you, say, pushed on a domino, it would cause all the other dominoes to fall on the chain. Now, of course, we're not just interested in how birds produce complex motor sequences, we're interested in how humans produce complex motor sequences. Unfortunately, it turns out that the structures that have been identified in the bird that are important for the song production have exact parallels and homologies in mammalian systems, including humans. It includes a loop between the cortex and the basal ganglia that's important for this production and learning in mammals. Now, how do we understand how some sort of dysfunction in this circuit could lead to a complex disorder of behavior? Well, it turns out that often the disorders of circuits come about through disorders of their synapses, the things that connect the neurons to each other. And Guoping Feng, who is one of our most recent recruits to the McGovern Institute, we recruited him here from Duke and now occupies the Poitras chair here in the department, has been studying these synapses for some time, and he and his colleagues have established that the synapse, even though it's a very tiny structure, has over 1,000 proteins. You can map out what those proteins are, And Guoping has been studying how dysfunction in one of these synaptic proteins could lead to a serious psychiatric disorder. And he's doing this in animal models. Now you could ask, exactly how does one create an animal model of a human psychiatric disorder? If you think particularly of the mouse as our common model, and then think about the kinds of symptoms that we see in a psychiatric disorder like mania, where you have inflated self-esteem, more talkative, flight of ideas, feelings of worthlessness, recurrent thoughts of death. You think, how easy is it to know if the mouse has feelings of worthlessness or an inflated sense of self-esteem? And it's true, it's a very difficult problem. And so we tend to focus on very simple, measurable behaviors, which are nonetheless components of more complicated disorders. So Guoping has been studying the impact of mutations in a gene that leads to a protein known as SAPAP3 in the synapse that is localized to that basal ganglia structure that I just told you about, the one that's involved in both learning of birdsong and production of complex sequences in humans, and is known to be dysfunctional in disorders like Parkinson's disease and many other psychiatric disorders. And he's been studying how a mutation in that gene could affect the circuitry in this structure in a way that could produce a specific behavior linked to a disorder. And what he finds is that the specific mutation in that pathway leads to something that looks like obsessive grooming in the mouse. You can see here the mouse is just grooming, which has a uncanny resemblance to the kinds of excessive grooming behaviors you see in people with obsessive compulsive disorders. And he can establish that it's the dysfunction in just that particular circuit with that gene by using gene therapy to restore the gene to that animal just in that circuit and show that in fact it cures the disorder in that mouse. This is very encouraging because the mouse has had this mutated gene since before birth, and yet even this treatment in the adult is enough to cure the disorder. Inspired by this work in the mouse, Guoping went to his clinical collaborators studying humans with OCD and did a search for genes in OCD that would include mutations in this SAPAP3 gene that he identified in the mouse to see if it might possibly contribute to the disorder in people. And indeed, they found that in a certain proportion of cases, humans also have a mutation in that same gene. It's not the only disorder that's going to come about as a result of dysfunctions in these synapses. In very recent work, Guoping has been studying the next protein down in the sequence that's called SHANK. And what he has found is that if you take out this SHANK gene in animals, you get a disorder that resembles some aspects of autism in people. It's particularly what you get as a disorder in social behavior. You put two normal mice in a cage, they interact nicely, sniffing each other and so on. But if you take two mice with this mutation in a cage, you can see that they will avoid each other. Again, resembling some aspects of the social dysfunction in autism. Now, we don't know what aspect of the basal ganglia circuitry would lead to some kind of social problem like this. But this is obviously a key area for us to study if we're going to make progress in eventually having therapeutics. Now, I told you that it's an imbalance in this basal ganglia circuitry. It comes down to an imbalance between excitation and inhibition in certain parts of that circuitry that give you these kinds of disorders. And Yingxi Lin, another one of our new faculty members, has been studying this balance between excitation and inhibition in a somewhat different way, but also with very interesting implications. Yingxi's work has established, of course, you have to have this balance between excitation and inhibition, or the brain will have a serious dysfunction. All the neurons in your brain have excitatory and inhibitory synapses, and we know that if you have an excess of excitation, neurons will try to increase their number of inhibitory synapses to try to bring down the excitation to put the cell back into balance. And Ingshi and her collaborators have identified the gene that regulates this activity-dependent growth in inhibitory neurons. It's called NPAS4. And as you might expect, if you take out this regulation, you get a problem like a broken thermostat. Now, if you have a thermostat hooked up to your air conditioner and the room gets too hot, the air conditioner kicks in, turn on the air conditioning, and bring the temperature back down. Well, if you don't have NPAS4 and you get too much excitation, there's not enough inhibition to balance it out, and what you get in mice is seizure disorders. It turns out that many people with autism also suffer from seizure disorders, and a very interesting link to autism came about in a study in which Ingshi was a collaborator, in which it was found that a gene that is linked to NPAS4, is regulated by NPAS4, actually is a vulnerability gene in autism. And a further interesting clue of a link is that in the most recent work that Ingshi has done, she has found that not only does this gene regulate the formation of inhibitory synapses, but it seems to be a regulator of learning and memory in a particular neural circuit that's important for memory formation in people. And she's found that if you mutate this gene, you get this very specific learning impairment. So here you have a gene that has this diverse effects, increase in seizures, learning disorders, and you can't help notice the similarity to some of the impairments that are seen in autism. And this is, again, something that's being actively pursued in Ingshi's lab. Now, we're not just interested, of course, in these animal models. We also want to understand neural circuitry in people. And we have faculty in the McGovern Institute that are studying human subjects, including John Gabrielli, who was recruited here from Stanford and is now head of the Imaging Center. And John and the many other people who are using the Imaging Center are mapping out circuits important for all kinds of high-level cognitive functions, perception, attention, memory, and so on, as well as mapping out circuits that may be involved in serious disorders, such as schizophrenia and bipolar disorder. You can also use this MR technology for establishing the connectivity between these different circuits. You can now use sophisticated analysis methods to actually find the axon bundles that connect the cells interacting across all these different brain systems. Once you have a sense of that circuit that's involved in a behavior, then you can think about manipulating it. And in very recent work, John and his collaborators have shown that you can use MR itself to provide feedback to the subject that will enable them to regulate their own brain activity. And this has been tested in a case of pain, where they took subjects who suffer from chronic pain, they put them in the scanner, and they localized the part of the pain circuit that was known from other mapping work, and they showed the subject how much activity they have in their own pain center. And the subject was given training to lower the amount of activity in that structure. And as a result, this lessened their feelings of chronic pain and extended beyond the time when they were in the scanner. And now they're going to try the same kind of approach in depression, where they're going to try to get subjects to learn to regulate activity in their nucleus accumbens, which is also implicated in the depression circuit. Now, MRI and these techniques, they give us really a unique view into neural circuitry in people, but not the kind of view that we get from our animal studies, and certainly not the kind of view that we would really want to have, and particularly because MRI is usually averaging over seconds, whereas our brain states are changing in milliseconds. But fortunately, there's a technology that we will soon have here at MIT. It's being, in fact, built into the Imaging Center right now, known as magnetoencephalography, that's going to give us that temporal picture of brain activity. And this has come about through the support of generous donors, some of whom are in this room right now. And using MEG, which I should also point out was developed here at MIT 40 years ago by David Cohen, And it's taken 40 years for us to have a state-of-the-art MEG imaging center here at MIT. But we have it now, or will have it now, I should say. And MEG is going to give us that kind of picture of dynamic changes across the brain that we know are important in all these complex behaviors that we want to understand. Another limitation of MRI is it was based on technology that was developed 20 years ago. And that might lead you to ask, so where's the technology that's going to put us where we need to be 20 years from now? And I think the work of Alan Jasanoff here is giving us a glimpse of where that could be in the brain imaging field. What Alan's doing, he's using the latest techniques for genetic engineering and directed evolution to create molecules that can act as sensors in the brain for important neurotransmitters and other important brain proteins that are involved in these kinds of disorders I've been talking about. And the molecule he's developed most recently is one that is a sensor for dopamine. And you can see here in this image through a rat brain where the red blob here shows you dopamine release in the basal ganglia. Again, that part of the brain that I've been telling you about. We've done all this work on neural circuitry. He can now image the dopamine release in that structure. Dopamine, of course, is the key molecule implicated in Parkinson's disease, is also implicated in schizophrenia and other disorders. Now, these are early days. These are coarse images. This is not ready for prime time, not ready for use in people. But I think what this is pointing us to is where this field is going in the future, where I believe over the next decade, we are going to be able to put together all this kinds of technology that I've been talking to you about today, the technology for understanding neural circuits, for understanding the molecular biology, from manipulating the circuits with light, targeting different signaling pathways in cells. And we're going to be able to link that up with technology that's going to give us readouts of comparable kinds of measures in human subjects. And we are going to truly lay down the foundation for what it will be the new therapeutics of the future. And that's my own goal for what we can contribute towards in the McGovern Institute over the next decade. At this point, I'd like to say thanks. I would like to thank the many students and postdocs working in the McGovern Institute who actually do all this work in the lab. They're the ones in there every day, running the experiments and so on. And then when they finish here, they go off and set up their own labs in universities, increasing our worldwide impact. And this is just showing pictures of some of the people that have left the McGovern Institute in the last couple of years and set up their labs around the world. And of course, I'd like to thank our founders, Pat and Lori McGovern, for having the vision to create the Institute and supporting us. And their active involvement with the Institute, which has inspired so many others to contribute to our work. And many of those people who have been contributing to our work are here in the audience today. And our staff, our McGovern Institute staff, without our staff, the McGovern Institute would not function for one day. And certainly the event that you're in right now would not be taking place and truly need to be acknowledged. And then thinking about how I should end this wrap-up of the McGovern Institute, what would be the parting words? I couldn't actually think of anything better than some words that were spoken by one of our founders, Lori McGovern, that appear at the end of our McGovern Institute video. And rather trying to paraphrase Lori, I'll just give her the last word.
I believe we give people hope. And when there's hope and not defeat or despair on people's mind, progress will prevail and solutions will be found and neurodegenerative diseases will be cured. I just know that.
Thank you for your attention. Thank you for coming. Since then, the McGovern Institute has been in operation a total of 10 years. So this would be a good time for us to reflect on why are we here? What are we trying to accomplish? And what sort of progress are we making? That brings us to the mission of the McGovern Institute, which is a twofold mission. The first is to understand the mind and brain, and the second is to apply that knowledge to understand what goes wrong in a variety of serious brain disorders. This is truly an astronomical problem. There are as many neurons in our brain as there. Welcome to the 10th anniversary of the McGovern Institute. So, the McGovern Institute. It all got started with the vision of Pat and Lori McGovern to create the McGovern Institute for Brain Research at MIT. That led to the construction of this wonderful building complex, and which opened 5 years ago. In fact, we had our opening celebration almost 5 years today, and I recognize many people here today were at the opening celebration 5 years ago. 1 in 4 families in this room will have a brain disorder, will be affected by a family member with a brain disorder of some sort. This could be a psychiatric disorder, a disorder of childhood such as autism, a disorder of the elderly, such as Alzheimer's, dementia. And I don't need to tell this group that the treatments we have for these disorders are woefully inadequate. But it's not my goal here today to depress you, but rather to tell you why we have so much hope about progress over the next decade. And our hope springs from what I would say are 3 revolutions that have taken place in science in recent years. And those revolutions have been in genomics, in systems neuroscience, and in human brain imaging. And they have depended in part on very significant advances in technology. So, how are we making use of these new opportunities to drive progress here at the McGovern Institute? Well, first, as I told you, we're pursuing a dual-fold mission, which is to support basic research as well as research that we call translational, or with an eye towards explaining or helping in a serious mental disorder. And we put this all together in the same place. Secondly, we encourage what we call a culture of entrepreneurial research. So there are stars in the Milky Way. And it's the interaction of these billions and billions of neurons that lead to who we are. It leads to the human mind. It leads to our shared thoughts and dreams and aspirations. It leads to the ability to learn and remember. It leads to our innovation and creativity. Unfortunately, it also leads to conflict and sometimes leads to reconciliation. And when it goes wrong, when the processing goes wrong, it can result in a serious brain disorder. And statistically,